to the body's frontline defense. Here in the bloodstream, a vast river of red blood cells carries oxygen through plasma. But danger lurks. A bacterial pathogen slips in through a cut in the skin, which, as it invades neutrophils, the immune system's rapid responders patrol vigilantly, guided by the chemotaxis as they follow chemical signals like cytokinesis released from the injury site. Receptors on the neutrophil surface bind these signals, activating internal pathways that propel the cell toward the bacterium. Deep in a capillary, the neutrophil receives activation cues from damaged tissues, releasing DAMPs, damaged associated molecular pattern. Signals bind to toll-like receptors on the surface, triggering an intralectural cascade via the NFKB pathway that mobilizes granules for action. The nucleus reshapes, preparing for diapedesis, the dramatic sequence through the vessel's wall. Now the neutrophil closes in. First comes opsonization. Antibodies or complement proteins coat the pathogen, marking it for destruction. Pseudopores extend surrounding the invader. The membrane invaginates, forming a phagosome that seals the bacterium inside. Zooming deeper, the phagosome fuses with lysosomes to create a phagolysosome. Proton pumps acidify the interior, while digestive enzymes like lysozyme and protozoa flood in. Reactive oxygen shuts the bacterial cell wall. An oxidative burst unleashes superoxide radicals. Debris is expelled. Finally, the neutrophil may undergo NETosis, releasing chromatin webs. Neutrophil extracircular traps to insert even more invaders. Monocytes now join the fight, drawn by CCL2 chemokinases from the inflamed tissue. They roll along the endothelium using selectins, then achieve firm adhesion with integrins before transmigrate out of the blood vessels and differentiating into macrophages in the tissue. Macrophages, the great tissue guardians, engulf apoptotic cells and debris. They recognize phosphatidylserine, each me signals, extend large pseudopods in a zipper-like embrace and digest everything inside powerful lysosomes. Inside the macrophages, antigens are processed. Degradation in endosomes produces peptides that load onto MHC class II molecules. Vesicles carry them to the surface, ready for presentation to T cells, the crucial bridge to adaptive immunity. In the skin, dendritic cells act as sentinels, capturing antigens. Upon maturation. They upregulate co-stimulatory molecules like CD80 and CD86, then migrate through the lymphatics to the lymph nodes. There, in the lymph node, the dendritic cell present antigens on MHC to native T cells. The T cell receptors bind the complex co-stimulation through B7, CD28 follow, signaling via ZAP70 ignites the T cell into action. Activated CD4 plus help by T cells differentiate into specialized subset Th1, Th2, Th17, guided by the cytokinases such as IL-12. Transcription factors like T-bet switched on and waves of cytokinases recruit more allies. Meanwhile, CD8 plus cytotoxic T cells detect viral antigen displayed on MHC class 1 of infected cells. They undergo rapid clonal expansion, loading perforin and granzyme granules, priming for the kill. The cytotoxic T cell talks with the target, forming a precise immunological synapse. Perforin punches pour in the membrane. The enzyme rush inside, triggering cascades that drive the infected cells into apoptosis. The target cell blebs, fragments, and dies. In the lymph node follicles, native B cells encounter antigens, BCR cross-linking triggers internalization and processing for MHC2 when they present to helper T cells, CD40, CD40L. Co-stimulation seals their activation. B cells are racing to germinal centers. In the dark zone, they proliferate violently, while somatic hypermutation defines their receptors. In the light zone, follicular dendritic cells select the highest affinity clones. The winners transform into plasma cells. Their endoplasmic reticulum swells massively to produce antibodies by the thousands. Class switching begins. IgM gives way to the more powerful Ig. These antibodies pour into the blood and tissue. Their Fab arms bind viral epitopes, blocking entry. Antigens tag pathogens for phagocytosis. They clump invaders together and trigger the complement cascade. In the classical pathway, C1q binds the antibodies of CDgen, igniting a domino chain that ends with the membrane attack complex, initiate pores that RAP open the pathogen's membrane and destroy it. Eosinophils arrive to battle parasites summoned by IL-5. They unleash major basic protein that tears into the parasite's tough cuticle and perform antibody-dependent cellular cytotoxicity. Basophils and mast cells triggered by IgE cross-linking explode, the histamine blood vessels dilate, walls become leaky, and fluid rushes and creating the classic signs of inflammation that summon even more white blood cells. Natural killer cells patrol constantly hunting stressed cells that have lost MSC class 1. They're activating receptors while releasing perforin and enzymes in a swift, innate execution of tumor or virus infected cells. Eventually, inflammation must resolve. Macrophages switch to anti inflammatory mode, releasing IL-10. They quietly perform a phagocytosis, clearing the battlefield of spent neutrophils. Tissue repair signals rise, blood flow normalizes, and peace returns. Yet the immune system never forgets. Surviving T and B cells become memory cells. Their DNA marked by epigenetic changes that allow lightning fast reactivation. They circulate through blood and lymph forever vigilant. The next time the same threat appears, the secondary response is swift and overwhelming. Memory B cells flood the system with high affinity antibodies and memory T cell activating hours rather than days. From the first neutrophil racing through the bloodstream to the final restoration of harmony. White blood cells orchestrate an astonishing symphony of detection, destruction, coordination, and memory. These microscopic warriors, neutrophils, macrophages, dendritic cells, T cells, B cells, all the rest and God every second of every day quietly protecting the incredible world inside us. Thank you for joining this journey through the immune system's wonder.